Welcome everyone. Thank you all so much for attending our OAI.edu live webinar series hosted by the OAI CORE Knowledgebase. I want to thank today's speaker, Dr. Michelle Yao, who is an assistant scientist at the Marcus Institute for Aging Research at Hebrew Senior Life and an assistant professor of medicine at Harvard Medical School. The title of today's talk is Genetic Data Resources in the OAI, a Primer. Following the presentation, we should have a few minutes for Q&A, so if you have any questions, feel free to type them in the chat, and we'll try to get to them at the end. And now I'll let Dr. Yao take over.
Great, thank you. All right, so today I'm going to go over some of the genetic data resources in the OAI. If I can get this to So the data that we have available, it's whole genome genotyping, which was originally obtained back in 2009, 2010. And they were obtained on most of the OAI participants using the Illumina OmniQuad 2.5 million array. This was part of an ancillary study called the Genetic Components of Knee Osteoarthritis, the GECKO study, which was led by Dr. Rebecca Jackson. It included over 4,000 individuals, most of which are European ancestry. We do have almost 1,000 African ancestry individuals who are genotyped, and they were genotyped as part of at the Translational Genomics Research Institute. So with these data, we had a number of quality control metrics that we used. First were the sample exclusions. So we excluded individuals who had call rates less than 98%. And I have the number of European ancestry and African ancestry individuals who met those criteria in the parentheses. And we also looked for mismatches between reported and genetically determined gender, as well as looking for second degree or higher relationships among samples to ensure that we had independent samples. Though nowadays you might just calculate a genetic relationship matrix and use that instead. But at the time we had just excluded individuals who had second degree or higher relationships with other samples in the cohort. We also excluded individuals with large chromosomal abnormalities. And there were a handful that met those criteria. Additionally, you'll note that what we've provided are these self-reported races. And if you look at the genetic data, there are a number of people who have a self-reported race that is different from their genetically determined race. So that is something that you would want to look into if you're doing genetic analyses in OAI. And depending on your question, you may or may not want to use the self-reported race. Typically for the genome-wide association studies, we've often corrected this and used their genetically determined race when splitting it by European versus African ancestry analyses. We also have SNP variant filters. And so we removed those SNPs that were duplicated. We also removed SNPs that had call rates that were less than 95%. And we did have a number of planned duplicate samples so that we could look at discordant genotypes. And where there were 2 or more discordant genotypes, we excluded those SNPs. And then it's up to you whether you want to impute these data or not. We've oftentimes imputed the data to the Haplotype Reference Consortium, and we also have imputed to the TopMed imputation panel. But it sort of depends on you, which panel you want to use, and which reference you want to use. So we've used these data initially in this publication back in I think it was 2013, where we looked at BMD-associated SNPs with knee osteoarthritis. And so we had taken our genotyping data in OAI, and we wanted to see whether the current genome-wide association study for BMD, and at the time I think this was from the GFOS consortium, and we wanted to see whether those BMD SNPs were associated with OA. We found a number of SNPs that were associated with OA, and these were SNPs that were associated with high BMD, increasing the risk of OA. But when we put these SNPs into a polygenic risk score, we found that it was not associated with OA risk. So it was just specific loci that we found that were associated with OA. We then did a genome-wide association study with structural knee osteoarthritis, including a number of other cohorts, including the Johnston County Osteoarthritis Initiative, the MOST multi-center osteoarthritis study, and the GO cohort, genetics of OA cohort, genetics of generalized OA cohort. And so here what we identified was one novel locus and then two that were already known. So this was the GDF5 locus and FTO. And so at the time we had decided to split it by European ancestry versus African ancestry. So we published two separate GWASs for that. And we had another one looking at genetic determinants of knee osteoarthritis in African Americans. And we found one novel locus, and interestingly, didn't find any of the European ancestry variants that were associated with knee OA in African ancestry individuals. And most of this was just driven by a lot of allele frequency differences between the two populations. And we also had quite a small sample size for this. So it made it quite difficult to find genome-wide significant loci in this effort. But these data have been deposited into dbGaP, and you'll see that you can request access for these data. And there's a description here about the data, and note that this is controlled access only. And so you have to have meet certain requirements in order to receive the data. So the data access committee reviews your application. You have to provide the purpose of your research, why you're using the data. You have to demonstrate that you have IRB approval to use these data, and also agree to the dbGaP data use certification. And I'll go over that a little bit more in the next few slides. One thing to note here is with the phenotypes in dbGaP, we only have the age, gender, and race. So you will have to go to the OAI knowledge base to pull the phenotypes, but then you can link these genetic data to the phenotypes in OAI. And so a note about this data access piece of it. So you do have to sign a data use certification agreement, and starting in 2025, they made it a little bit more stringent in terms of the amount of security you need in order to store this data. So any server that you plan to put these data on have to meet the NIST SP 800-171 criteria. And so this is essentially a federal guideline that gives you the requirements that you need to protect unclassified, controlled unclassified information within a non-federal system or organization. And it's just to ensure participant safety and privacy. And it's a pretty stringent criteria. I think a lot of institutions have had some trouble meeting these criteria. So do check with your IT to make sure that your server meets this criteria. And also note the data cannot be put on your personal computer. It has to go to a secure location. Okay. So now fast forward a few more years, we contributed OAI data to the largest effort to date, looking at genetics of OA as part of the Genetics of OA Consortium. And so this effort identified almost 1,000 loci now associated with OA, of which about half of them were novel. And so what's really, really exciting about this effort is that even though we were a teeny tiny piece of it, I think because of the depth of phenotyping that is available in the OAI, it gives you the opportunity to be able to look at these variants identified in this large GWAS in more detail. And so again, you can try to refine OA phenotypes using the Go Consortium GWAS data. And so one thing we've done is look to see whether those variants identified in the Go Consortium GWAS, how they're associated with structural versus pain phenotypes. We found that there are much stronger associations with pain phenotypes than with structural phenotypes. Additionally, OAI provides the opportunity to look at a number of imaging phenotypes that are not available in other cohorts or biobanks. And so there you can take the Go Consortium GWAS variants and look to see how those are associated with novel MRI phenotypes, for example, or any other imaging phenotypes. I think a number of people are doing machine learning on some of these images, and there could be some novel phenotypes derived from those efforts. I think there was also an effort to look at genetics underlying knee bone shape as a novel phenotype. And so I think there are a number of opportunities to really dive into the Go Consortium GWAS to really refine those phenotypes that are associated with the variants that are identified. The other novelty to OAI is that you have longitudinal data. So this gives the opportunity to look at associations, genetic associations with longitudinal change in OA outcomes. And in fact, we did this many, many years ago, and we had looked at joint space narrowing progression, osteophyte progression, KL progression, and progression to joint replacement. And what you'll see in these Manhattan plots is with the OAI data alone, you can't really find anything that's genome-wide significant. There's a number of peaks that look suggestive or look promising, but it's really difficult to just do a GWAS in the OAI and be able to identify anything in it. So I think really the novelty of the OAI genetics data is to really refine those genetic variants that have been identified through these, huge efforts, and be able to dial in on those variants that could be associated with progression. Another place where you can use these genetic data are in polygenic risk score associations. We've used this to look at OA risk stratification. Using the Go Consortium data, you can come up with a number of polygenic risk scores. You can look at polygenic risk scores for any OA, hand OA, knee OA, any phenotype that they've looked at in the Go Consortium GWAS. You can derive a polygenic risk score from that and apply it to OAI and look at how those polygenic risk scores perform, predicting OA outcomes in the OAI. Additionally, we've used the polygenic risk score approach to look at relationships with other traits through genetic risk. So other traits have large GWASs as well. And so we've been able to derive a lipid genetic risk score, for example, an Alzheimer's disease genetic risk score, to look at how those genetic risk scores associate with OA in the OAI. So I think this is a place where you could really sort of harness what is available in OAI with the genetics data and really combine what's known about genetic risk for OA and other traits in order to sort of refine and dive into the deep phenotyping available in OA and see what's associated there. And of course, for any genetic analyses, you're always looking for validation. So it's hard to just do an analysis in one cohort and get that published. And so there are a number of cohorts that have similar data, the genetics data, and the OA phenotypes available. And those cohorts are the Framingham Heart Study, Johnston County Osteoarthritis Study, and also the Multi-Center Osteoarthritis Study. And so I just name a few, but I think there are a few more. But if you're looking at North American-based OA cohorts, these would be the primary three, I think. And finally, I think there are opportunities to integrate these genetic data with other multi-omics data. So there is the musculoskeletal knowledge portal where we're working on adding a number of functional genetics data into this portal, as well as GWAS data from all musculoskeletal phenotypes. And what you can do is if you do a GWAS of your favorite trait in OAI, you can upload those data to the musculoskeletal knowledge portal. And within it, there's a number of gene expression data, single cell data, and you can integrate those findings from your GWAS with single cell data, what's known about gene expression data, epigenetics data, and I think there's a tool within the musculoskeletal knowledge portal for you to be able to integrate those data. Or if you want more simply just to look something up, you can do that as well. And this portal actually sits within an umbrella of a whole bunch of other portals. So there's also a metabolomics portal. And so there are ways to be able to link what you're finding in OAI to what's happening across other systems and across other genetic functional data. And I think those are just a few of the ideas that come to mind in terms of using the OAI genetics data, but there are a number of other possibilities. And I think a question is, are there other omics data that are available in the OAI? And I think there are, but I have not seen them on a publicly released platform. And so if you know of any ancillary study data that would like to make those data publicly available, please let me know. I think this would be a huge benefit to be able to integrate the genetics data with other omics data that people have collected. And so I think there's more to come in terms of integrating or collecting more omics data within the OAI. And I think also looking forward, what we want to do is be able to aggregate more omics data in the OAI. So collect more omics data and also be able to combine these data with other longitudinal cohorts in a mega-analysis. So just figure out a way to just combine everyone's efforts into one to be able to yield more from the data that everybody is collecting. And then also right now, the OAI knowledge base and the musculoskeletal knowledge portal are separate entities, but I think we're thinking about how we can integrate these two knowledge bases in order to benefit the musculoskeletal community. On the one hand, OAI has deep phenotyping for OA that's extremely valuable to the community. And then the musculoskeletal knowledge portal has a number of genetics data and functional data that's available in musculoskeletal tissues. We're working on collecting more single cell data on tendon, cartilage, bone, anything that's relevant to a musculoskeletal trait. And finding a way to integrate these two, I think, would be ideal to enrich both resources. And so I think, in summary, there's a number of possibilities for using what's already been generated into OAI, but I think there's more to come. And so if you have any questions, please let me know. I will be at ORC, so if you want to connect, just find me. And then if you're having trouble with the OAI stuff, let me know. I can try to help you out. But anyways, thank you for your time, and I will end there.
Great, thank you so much, Dr. Yao, for that excellent presentation. We have a few minutes for Q&A, so if you haven't done so, feel free to ask a question in the chat. And I'll get us started here. Do we know if the people who have GWAS data are similar to the overall OAI study characteristics?
They should be, because we have genetic data on most of the OAI participants.
Great. Another question, are OAI genetic data continually updated?
So no, they are not. This is based on a one-time effort to collect genotyping data on these participants. So it's all based on the 2.5 million array. But what you can do is impute these data to different reference panels. So in that sense, you could update the genetic data, but that's just updating the imputed data. But the raw genotyping data itself is not being updated.
Can the GWAS data be linked to the publicly available OAI data? And if so, how? For example, do you merge the files by the public OAI ID?
It should be by the public OAI ID.
Okay. Great, thank you. I don't see any other questions in the chat right now. Oh, actually, we just got another question. Someone asked, can you say more about the pain analyses?
Was there something specific you were looking for? So we had looked at a polygenic risk score with various OA outcomes. So looking at its association with structure and its association with various pain phenotypes. And we really found that these polygenic risk scores were very much driven by pain, and the associations were much stronger for pain than they were for structure. Luckily, a lot of the associations, if you looked at the knee OA polygenic risk score, it was associated with knee OA. If you looked at the hand OA polygenic risk score, it was associated with hand OA. And so they intended to be joint specific. But if you look at just its effect size, I think pain, for some reason, a lot of these polygenic risk scores just had an underlying association with pain.
We have a follow-up to that. They said, it's interesting that the genetic markers are strong versus biochemical markers require more markers to identify pain phenotypes. Where are your pain results published? Very interesting.
It is published on my desktop, and I will get this paper out, I promise.
Another question, does OAI have omics data?
Does OAI have omics data? So I know that there are ancillary studies that have either collected these omics data in a subset of OAI individuals. I don't think it's been done on a large scale in the OAI. So I think there is methylation data that has been collected. I think Francisco Blanco's group has done a number of I think genetic analyses using haplotyping and methylation data. So those are the ones that I know about. There might be more. I've not seen it. Additionally, I think what would be really helpful is if these data were publicly available for others to use. And I've not seen those publicly available, but I could be wrong. But I've not seen it.
Okay, we have one last question before we end. How do you curate knee MRI scores like worms, and is there a way to link them to omics data?
Yeah, so I would probably punt this to Jeff. And so I think what you're talking about are the phenotypes that are available in the OAI knowledge base. And so I think anything that's in the knowledge base should be linkable to the genetics data. They may not be linkable to the methylation data that others have collected, but certainly they should be linkable to the genetics data.
Yes, thanks, Michelle. Any of the MRI data that is publicly available through the NDA website would be linkable with the GWAS data. It's similar to using other MRI scores. It's important to think about which read projects they were derived from, but that's certainly something too that the knowledge base is willing to sit down with you and talk through the feasibility and which read projects can be merged, or caveats to thinking through how to do that and what statistical and analytic considerations we need to have about doing those merges. But they can be merged with the publicly available clinical data as well as the GWAS data. And to Michelle's point too, there are some nested studies within the Osteoarthritis Initiative that have done omic metrics. For example, there was a case cohort of 603 people who had plasma metabolomics completed. One of the goals of the knowledge base is to engage more of these investigators to help get some of this data publicly available. So hopefully you'll be seeing that in the near future.
Great, thank you. That's all the time we have for today. Thank you again, Dr. Yao, for that excellent presentation, and thank you all for joining us today. And as we wrap up, I want to invite you all to our upcoming webinar on May 11th with Dr. Charles McCullough, who will be talking about data analysis strategies for the OAI. It's going to be another great session, so we hope you all can make it. And I sent a link to register in the chat along with other links to our website and the webinar survey. So that concludes today's webinar. Thank you all again and have a great rest of your day.
Thank you.
Thank you.